Thank you all for coming to the Memory and Aging Project Knight Alzheimer's Disease Research Center Participants Meeting. We're delighted to welcome you. My name is John Morris, and I have the real honor of being the director of the Knight Alzheimer's Center and its Memory and Aging Project. So on behalf of all of my colleagues, we're so delighted to see so many of you come for this meeting. Now, just some housekeeping details. If you have a smartphone or any type of cellular phone, will you be certain that it's off or on vibrate? And if you exit the ballroom here, the restrooms, I think, are off to the left. I believe the coffee and food stay out for a little while. Did you enjoy the breakfast? I told some of you already, but I was cooking all night long. So. Now let's see if I can do this. Yep, okay. Oh, good. Look, first slide. Turn off your cell phone. So here are many of the faculty and staff of the Alzheimer's Disease Research Center, along with several of our supporters. It is a terrific group of individuals, investigators, staff, and we are really pleased to be able to work together in a coordinated and integrated fashion on this terrible illness, Alzheimer's disease. Now, many of the staff and faculty are here today to give you our appreciation for all that you provide to the Memory and Aging Project and Knight Alzheimer's Disease Center. We cannot do it without you, so we give you a really great note of appreciation for your time, commitment, and your dedication to the cause. Now, as long as I have this slide up, I hope I don't overlook too many people, but I'm going to ask different members of our faculty to stand. Dave Holtzman, who is the Associate Director of the Alzheimer's Center and the Chair of the Department of Neurology, so Dave will stand over here. It's a long list, people. I think we'll save applause till the end. Oh, you can certainly applaud Dr. Holtzman. He's my department chair. But now let's hold the applause until the end. Dr. Virginia Buckles is the executive director of the program, as is Krista Mulder. So Virginia and Krista are standing there somewhere. Lenice Cummings-Vaughn is a geriatrician working in the clinical core. And Lenice, are you standing? Okay. Lenice, thank you. Ann Fagan is the biomarker core leader. Where is Ann? Ann, okay, great, thank you. Nupur Ghoshal is a clinician in the Memory and Aging Project. Nupur is over here. Jason Hasenstab leads our psychometric unit. There's Jason. And Joy Snyder is one of our clinicians in the Memory and Aging Project, and she also is the head of the training program for all new neurologists, the neurology residents. So let's give all of them a great round of applause. We also have additional individuals, so again, hold your applause until the end. We have two physicians who are working with us as clinicians, Dr. Finay Dadi. Finay, you're standing up. And Dr. Renata Reimers. Renata is here. In the back was Bo Ansis. Is Bo still here? Where's Bo? Over there. One of our neurology faculty and imager, so you'll hear from him. Sarah Hartz from the Department of Psychiatry. Where's Sarah? Sarah's over here, okay. Brendan Lucy, who, along with Dr. Holtzman, is running our sleep studies. Susie Stark from the program on occupational therapy. Where's Susie? Way over here. And we have international scholars working with us. From Japan, we have Ryoko Ihara. Ryoko is here. And from China, Hongban Lu. Hongban is standing up as well. So let's give them a round of applause. From the Alzheimer Association, Cheryl Kinney is here. Hold your applause. Cheryl, are you standing? Yep, thank you. From our African American Advisory Board, the founding chair in the far back, Norman R. Say. Norman, you raise your hand. And we also have Sally Simmons in the far back. Norman and Sally, you couldn't be further apart, could you? And Ron Gregory and Reverend C. Jessel Strong. So let's give them a round of applause. All right. So I'm going to bring you up to date on how we're organized and how we operate. So all of you, the participants, the volunteers, are evaluated at our office that we call the Memory and Aging Project, or MAP. The participation and the procedures have to be supported by grants that we get from the National Institute on Aging. We have 4 large grants. One is actually called the Alzheimer's Disease Research Center Grant, but others are called Healthy Aging, Senile Dementia, the Adult Children Study, and the Dominantly Inherited Alzheimer Network. And you can see the years where those grants were first funded. So the Healthy Aging and Senile Dementia began in 1984. But all of those grants support your participation in the Memory and Aging Project. Now, the way it works through the National Institute on Aging, if we're successful in being awarded the grant, they'll give it to us for a 5-year period, a budget period of 5 years, at which time we must apply for renewal of the grant. And this is a competitive process. It certainly is not guaranteed. We have to demonstrate that we made progress in the first 5 years, that we have more to do in the next 5 years, for which we're requesting funding, and that we have the capacity to do it. And then a committee of our peers decides whether to award us the grant or not. We have been extremely fortunate. Since 1984, all of these 4 grants not only were funded initially, but every 5 years have been renewed. Currently, the Adult Children's Study Grant is under review for its renewal period for another 5 years. So that's the way we work. Funding for your participation comes from the National Institute on Aging, part of the National Institute of Health, part of the federal government, ergo taxpayer dollars. That's what this is from. All right, we have a large staff, a large faculty, as you have seen. We've added individuals. I won't read them all here, but you can see who has been added to the team. And some individuals have gone on to other positions or have retired. So you can see the names of the individuals here who have left. One person in particular is leaving after 17 years of working in the Memory and Aging Project, and that is Doris Jones. So Doris, will you come up to the podium, please? So Doris is the face of the Memory and Aging Project, the first person you see when you walk into our office. And it's a real bittersweet moment for us to have to say goodbye, but I have to tell you that, keep coming, Doris, keep coming. I have to tell you that she has really a promotion. She's going on to another position that really is a tribute to her and all that she has done. Come on up, Doris. I'm not going to try to pin it.
Okay, I'll take it.
Okay, you might want to say.
Thank you. Oh my gosh, this is really a surprise. Oh, all I can say, because I don't want to take up a lot of time, is thank you. You all have made, this is the first time Jennifer Phillips got a picture of me in 17 years. And I can't duck and hide this one. But thank you. You all have made my job such a pleasant journey, and I really appreciate it. I appreciate your time, because we all do. I say this as a whole, as a part of the family. We all appreciate it because we know you give up our time, give up your time to come see us. So thank you. It has been really a good journey. Thank you so much. You know what, Dr. No. He just whispered this in my ear, and he didn't have to say it, but yes, the leadership at the Memory and Aging Project is phenomenal. Thank you. And he knows this because I've always put it in my email how much I appreciate what they do for the project. Because the leadership there, they do put a lot of time and effort to keep this going, and I appreciate that. Thank you.
In the past year, we've lost two individuals who are very close to us. One is Pam Millsap, a longtime nurse clinician, and she passed away just about a year ago. And then more recently, the current chair of our African American Advisory Board, Ida Goodwin Woolfolk, died at the end of March. Both Pam and Ida were really terrific contributors to the program, so we miss them very much. All right, highlights of the Memory and Aging Project and Knight Alzheimer's Disease Research Center. The 10th Norman R. Say lecture was held in October, and we had Richard King from the University of Utah come and talk about imaging and Alzheimer's disease. The first award of our grant, the Alzheimer's Disease Research Center grant from the National Institute on Aging, was in 1985, so last year marked the 30th anniversary. And we had a celebration at the Chase Park Plaza Starlight Room that was extremely interesting, fun, and poignant all at the same time. And I mentioned there, I don't ever want to have a 60th celebration because that means we haven't conquered the disease. So I hope we don't have to have these celebrations too much longer. We had an international symposium named in memory of the founding director of the Alzheimer's Center, my mentor, Leonard Berg. That was also held in October. Busy month for us. Coming up next month in Toronto is the Alzheimer Association's International Conference, the end of July, and we're going to be well represented from the Knight Alzheimer's Disease Research Center with over 40 presentations. And they run a gamut of topics, and you'll hear about some of those topics later today. This is a photo from the 30th anniversary. Dr. Snyder is in there. Some of you will recognize Mary Coates, Judy Leipoltz, Amy Buckley, and Norma Urani. The highlight of the 30th anniversary, I have to tell you, was I got my own bobblehead doll. I will be available after this meeting with my bobblehead for photos. We also asked the attendees to have a contest to see how much people knew about the Alzheimer's Disease Research Center, a trivia contest, if you will. And we're going to provide you with some of those trivia questions. Matter of fact, I think our team is distributing the clickers. Yes? Are they all distributed? Okay. So here's what's going to happen. Oh, by the way, this Mary Coates, not surprisingly, knew the most about the Alzheimer's Center, and she won the prize. Each table should have a clicker. You should pick a clicker captain for your table, because we're going to give you we're going to give you a few questions. We'll give your table 30 seconds to give an answer. Okay? Any questions? All right. Here's our first trivia question. Are you ready? Here we go. When was the first participant enrolled in the Memory and Aging Project? Okay, 30 seconds. All right, time is up. Here is the answer. There's how you answered, okay? And here is the real answer. All right. Next question. Next question. Are you ready? Here it comes. What did Dr. Holtzman promise his wife in exchange for her taking scuba lessons when they got married? All right, you have 30 seconds.
You close the polling, they can't vote.
I just hit it once, like you said.
It got closed. If you talk, I'll click.
All right. Ballroom dancing. Okay. Next question. If I told you we have 4 grants from the National Institute on Aging that fund our research, if you add up all of the money in those 4 grants for the 5 years of the budget period, how much is that for the Knight Alzheimer's Center total? 30 seconds. You don't hit it now to see.
No, you have to wait until they vote.
I thought they could see how the vote total changed.
No.
Okay. All right, here's how you voted. Here are your votes. I wish. Here's the real answer. All right, next question. We send out a newsletter twice a year to all of you. See what your memory is. What is the name of that newsletter? "30 seconds. Okay, here's how you responded. That's pretty good. Let's see what the real answer. All right, final question. For what sport is John Morris in the Ohio Wesleyan Athletic Hall of Fame? 30 seconds. All right, let's see how you voted. Okay, and here's the answer. That's it. All right. Thank you, Krista. Krista, are you collecting the clickers or Susie? Susie's okay. All right. More seriously, we are trying to defeat Alzheimer's disease. Our goal has shifted in the 30 years we've been in existence as an Alzheimer's disease research center to trying not simply to treat the illness once its symptoms are fully manifested, but to trying to prevent the symptoms from ever occurring. We call this preclinical Alzheimer's disease, no symptoms, but the brain changes of Alzheimer's are already present. And we think, if that's the case, that if the person who has those brain changes, even though they're perfectly fine in memory and thinking, if they continue to live, they will become symptomatic with Alzheimer's. So we want to try to detect those brain changes during life and start trials of experimental therapies that will halt the disease process and not allow the symptoms to occur, or at least delay when those symptoms do occur. So these are some of the procedures that we use to detect Alzheimer changes in people who don't have dementia, don't have the symptoms. We use a lot of imaging. MRI is an image, amyloid PET scan, tau PET scan, and we examine the Alzheimer proteins, amyloid and tau, in the cerebral spinal fluid, the CSF. These are the questions. Will all older people who have indications of the brain changes of Alzheimer's, have preclinical Alzheimer's disease, if they live, will they all develop symptomatic Alzheimer's disease? We don't know the answer. Can the period of changing from being mentally normal, cognitively normal, to becoming symptomatic with Alzheimer's, can we identify that area? And then finally, if we can identify that, we want to initiate drug trials of agents that might delay or prevent the appearance of symptoms. This is our major theme in the Alzheimer's Center. And we have new cause for optimism about some of these experimental therapies. As I say, there's a shift, not only looking at people who already have Alzheimer's dementia and trying to treat them, but trying to initiate these drug trials in people who still are cognitively normal, preclinical Alzheimer's disease. The very first of these trials began at Washington University under our dominantly inherited Alzheimer network. We call that DIAN. And part of DIAN has been developed into a trials unit. So you'll see the abbreviation DIAN Trials Unit. It's led by an ADRC faculty member, Randy Bateman. And it launched in 2012, targeting people who are cognitively normal, but they carry a rare gene mutation that means they will develop symptomatic Alzheimer's disease, Alzheimer's dementia, and at an early age, on average 45 years of age. So that was the first secondary prevention trial to launch. Other trials have launched subsequently, one called the Alzheimer Prevention Initiative, the other called the Tomorrow Study, and we are conducting at the Memory and Aging Project another study called the A4 study of older adults without any mutation, but who have signs of Alzheimer's disease in the brain. And that's called the A4 study, and soon we're going to add an A5 study for cognitively normal older adults. We're also not abandoning treating people who have symptomatic Alzheimer's disease. We're re-examining the way we've treated them in the past because we haven't had success in trying to treat them to date, but maybe new drugs or maybe different doses of current drugs can be contemplated. And you're going to hear from Dr. Snyder in today's program about these drug trials, so you'll hear more about it. I thought you'd like to learn a little bit about yourself. we see over 870 research volunteers in the Memory and Aging Project every year. Of those, about 500 are 65 and older. About 280 are in the adult children study. That begins at age 45. And this DIAN study, the one with the rare mutations, we see 92 individuals at Washington University. This is an international study. It is in the United States, Australia, Great Britain, Germany, Argentina, Japan, Korea. And so there are a total of 455 individuals in the DIAN study worldwide, but by far the largest is at Washington University. Here's the age range of our participants, 23 to 101. We're really studying this illness through the lifespan. Majority are women. If we include the DIAN study, 16% of our 871 individuals are African American, and we have one person who's participated for 30 consecutive years. Participants come from all over, including Idaho, California, and Maine. All right, so we are still looking for people to add to our research volunteers because some people move away or they might die, and we need to replenish the number of individuals. So we're looking for people who are cognitively normal, healthy older adults. We want 33 of those individuals to be recruited this year, and we're up to 12 who have come in. We're still looking at people who have Alzheimer dementia. We want 22 of those, but we only have 6. And in African Americans, we want 20 this year, and we only have 6. So we need you to help us in all of these areas. So healthy older adults, people with mild dementia, and in general, you see what we're looking for. People 65 and older, stable health, annual participation with a study partner, and at least in principle, willing to undergo our procedures, including that spinal tap, as well as the brain imaging. There's a phone number, but in the back, Andre Denny is standing and waving her hand, and she's particularly well situated because she's right next to Doris with her corsage. So Andre has the information for any of you to take, and if any of your friends or family are interested in at least learning more about enrolling, the information is back there. We really do need your help in order to have sufficient numbers of individuals to make the conclusions that we need to make. For those of you who are already in the Memory and Aging Project, we very much hope that you will continue to participate in all the things we ask. And by the way, we're appreciative that we ask a lot of you. We understand that. We ask a lot of you. We come in every year for a clinical evaluation, those memory and thinking tests. Everyone loves those memory and thinking tests, right? Then every 3 years for the imaging and the spinal tap. It's critical. We won't understand how those brain changes produce symptomatic Alzheimer's disease unless we're able to monitor them over time. So we really do need you to consider your continued participation. Tiffany, where is Tiffany? Tiffany is standing up in the back here. She's our scheduler. When she calls you, even if you cannot come at a certain time that she asks, please let her know, because she will call you back at a time that's more convenient. If you're moving, and many people downsize or move to be close to grandchildren, let us know your contact information. Now, once Tiffany schedules you for your evaluation, your assessment, your PET scan, we've noticed in the past several years that the cancellation rate has increased from 14% to 20%. Now, we know that have I said this? That we ask an awful lot of you. And it gets confusing. We appreciate that. But it's important for us to do our job, our mission, to conquer Alzheimer's disease through your participation. So baseline means the first time we ask you to do something. The MRI, 85% of you do, amyloid imaging, 72%, and spinal tap, 76%. You're the best group of individuals pursuing Alzheimer's disease in the world for all that you do. But you see, it falls off a little bit for lumbar puncture and when we ask you every 3 years. And now we're interested in the role of sleep. I mentioned that International Berg Symposium last October. It was looking at the interaction of sleep and Alzheimer's disease. What Dr. Lucy is doing and Dr. Holtzman is doing is understanding that relationship. Because you are so valuable and so committed, other investigators would like to have you join their studies. We call them affiliated studies. And at least 8 are currently active. And you see some of them listed there. A driving study, a study for tau, and looking at the ability not to remember what happened yesterday, but what we're supposed to do tomorrow. We call that prospective memory, okay? But all of this is more time and effort on your part. And we hear you when we call to ask if you would consider this. Some of you say, wow, another study? What study is this? I've been contacted a lot. And I'm full up on my calendar with all the studies that you're asking me to do. And there's a lot of research going on right now. It's true, there is. It's true, there is. So we're planning to try to understand better the burden we put on you with all of our asks and how we can reduce that burden and how we can provide you with things that you find even more valuable than coming in for your annual assessment. You're going to hear from Dr. Stark today about the burden study. Now, it sounds a bit convoluted, right? We're already acknowledging we ask a lot of you, and we want to understand that burden. What Dr. Stark is going to propose is we ask even more of you. We want you to fill out another survey. Dr. Hartz from the Department of Psychiatry is leading the charge to evaluate how we can get you your individual study results, because we know for some of you that's very important. And so we're committed to doing that. I mentioned all of our grants are reviewed by our peers every 5 years. Here are some comments from our reviewers. The clinical assessment follow-up deserves prioritization to bolster numbers. That's that cancellation rate of creeped up to 20%. The Knight ADRC is below the median in completion of follow-up visits. That cancellation rate again. Relatively low numbers of participants returned for spinal tap, lumbar puncture. Fewer than desirable number and frequency of lumbar punctures. So they notice these things. Now, frankly, I think those last 2 comments are unfair. Those reviewers don't have anywhere near in their own program the frequency of spinal tap that we do. So they're holding us to a standard they don't, but nonetheless, they notice these things. They notice this. So we appreciate that we ask much from you, but your participation is critical to help us advance the field of Alzheimer's disease and to ensure that the reviewers renew our grants. So we want you. Did you notice the change? All right, so I say the Memory and Aging Project leads the way in spinal fluid collection. It's true. Here are some other programs that are involved in spinal fluid collection, one in Australia, the one at Johns Hopkins, one at Wisconsin, one at the Mayo Clinic, but none of them have anywhere near the success rate that we do, and that is a tribute to all of you. And our external advisors, people come in every year, say we're at the top, and that's true, we are. Here's a recent comment from one of you. I'm in Dr. Morris's Memory and Aging program. This morning I had a lumbar puncture done by Dr. Ghoshal with assistance from Sushi. Sushi, where are you? Stand up. Sushi works with Dr. Fagan, and she coordinates the spinal tap program. I just wanted to tell you that they made the whole procedure almost pleasant. They were terrific in what they do. They made me feel very comfortable. You should be very proud of them, and we are very proud of them. Okay, I mentioned this already. Given the sophistication and advances in imaging and in spinal fluid analysis, we can detect brain lesions of Alzheimer's disease in living people. How many of you have heard you don't know for certain if someone has Alzheimer's disease until the person dies and the pathologist has to study the brain? No longer the case. We can do that in life now with these biomarkers. In life. So here are the brain lesions we're looking at. Amyloid plaques, abbreviated A and a Greek letter beta, neurofibrillary tangles, or tau. And we can evaluate these proteins, amyloid and tau, through these brain images using tracers. The original tracer was called PIB, but now we have others, including florbetapir, and the tau tracer is more recent, been coming along the past several years. And so we can combine these scans to do a brain autopsy during life. And I won't belabor this, but I just will try to show you here that this is the part of the disease that most people think about when you say Alzheimer's disease, the Alzheimer dementia stage. On average, it runs 7 to 10 years. But we think this dementia stage is the end stage of the illness. It begins 20 years before. These brain lesions build up over 20 years and eventually cause brain cells to die. That's a neuron is a brain cell. When they start dying, then you're on the road to developing symptomatic Alzheimer's. These are the ways we can detect this silent phase. And this is where we want to intervene before there's substantial brain damage and dementia symptoms are produced. Does that make sense to people? So Dr. Ansis will go over some of this, but here is tau imaging. A person here, no tau deposits in the brain, and that's what we see on this PET scan, and then it progressively builds up, and you see a lot of tau in this stage 5 and 6, and you can see a lot of the PET scan signal. Same with looking at the PET scan in a different way and looking at the amount of tau in the brain. The tau scan reflects the amount of tau in the brain tissue. Now, I'm talking a lot about imaging. I want to talk about radiation exposure because we ask you to undergo PET scans, which does expose you to some radiation. I want to explain this. So radiation is part of the natural environment. Earth materials and cosmic rays, even our food and water, all contain some radioactivity. The average annual dose of radiation from all sources is something called 2.4 millisieverts. I don't know what that means either. But the relevant number is 2.4. Now it varies. Not everyone gets 2.4. I mentioned the atmosphere, cosmic rays. The atmosphere protects us against cosmic rays. So if we live in a locale in the mountains, we have less atmosphere than someone who lives at sea level. So the person living in Montana will have more radiation from cosmic rays than somebody living in Baltimore. Even flying on an airplane puts us above area. We get more millisieverts per hour than if we didn't fly in an airplane. TV, computer screens, even that cell phone. So we're all exposed to radiation. Here are the natural and artificial sources. Remember I said 2.4 millisieverts per year, 2.4 millisieverts a year. Most of it comes from radon. That's an element that is derived from two other elements, thorium and uranium. It breaks down into radon. Radon decays very slowly. So it's the number one source of radiation. But here's the gamma rays, cosmic rays, food and water. So those are the natural sources. Here are the artificial sources, and number one among them is imaging. But nuclear tests, still some from Chernobyl, that meltdown, and nuclear power plants. Now, these are small, but they're detectable. So total per year, then, if you have imaging in particular, could be 2.8 millisieverts per year. I'll tell you right now, is that a lot? The regulatory agencies in the United States for people who work in radiology departments allow up to 50 millisieverts a year. Okay, safe. They determine that exposure up to that amount is safe. So on average, 2.8 millisieverts per year. But why are we interested in this? Well, radiation can cause cancer. We don't know how much radiation it takes, but on average, people who are more exposed to radiation are more likely to get cancer. We know this primarily from survivors of Hiroshima and Nagasaki, the atomic bombs in World War II. The way they do it is they damage the DNA in cells and cause the cells to proliferate. These are some of the cancers that have been associated with radiation: myeloma, leukemia, breast cancer, lung cancer, thyroid cancer, bone cancer, skin cancer. And it may take a long time to develop, decades. Lifetime risk of death per cancer. It's estimated in the United States it may be as high as 20 to 25%. Death from cancer. On average, 40% of us will develop cancer. Not all of us will die from it, but 40% will develop cancer. Men, slightly higher rate than women. And there are very vulnerable populations, including children, infants and children, and fetuses and pregnant women. But here's an example. A CT scan exposes people to radiation. If you were a 45-year-old and you had a full body CT scan, that's one way they diagnose people looking for cancer and other issues, then your additional cancer mortality is 0.08%. If you have that full body CT scan annually for 30 years, it rises to 1.9%, almost 2%. Still small, but not negligible. We can measure that. Now, why do people get full body CT scans? So they're looking for disease. So we have to weigh the risks and the benefits. Do we want to detect disease? If so, we might accept the risk of this. What about our imaging studies in the Memory and Aging Project and the Alzheimer's Disease Center? MRI scans do not have radiation. No radiation. PET scans do have radiation. And a CT scan, that we just mentioned, also has a small amount of radiation. We think that radiation is associated with developing cancer from the Hiroshima studies, but we monitor this very carefully. So medical imaging is the most artificial source of radiation exposure, and here are the millisieverts for a head CT scan or a PET scan. As I already mentioned, standards allow imaging personnel to have up to 50 millisieverts per year. All of our imaging studies at Washington University must be approved by the Human Research Protection Office and by our Radioactive Drug Research Committee. And we must ensure that the benefits, imaging the brain changes of Alzheimer's in order to identify people who might get an experimental therapy to prevent symptoms, outweigh the potential risks. And that we do everything that we can to minimize those risks. Dr. Benzinger is our imaging core leader in the Alzheimer's Center. She can't be here today, but she is very much committed to tracking and reporting every 3 months to that Radioactive Drug Research Committee and to the Food and Drug Administration nationally about our radiation exposure. And we track all of you and all of your scans in all of the studies to make certain that you're not exposed unnecessarily, and when you are exposed, it's at a level that we deem below the level of concern. I say all this because I want you to know that there are very few things in medicine that come without any risk, even taking aspirin. But this is something we take the radiation exposure very seriously, and so I wanted to share that with you. Now, at this point, we're going to hear from 4 speakers, and at the end of the presentations, we're going to have you use the cards on your table to write any questions that you might have, because at the end we'll have a panel discussion and try to answer your questions. And then we will close hopefully at 11. So the first presentation already mentioned, Dr. Susie Stark from the Program in Occupational Therapy is interested in how we can identify and hopefully reduce burden so that people who are volunteers are retained. Will you join me in welcoming Dr. Stark?
Thank you, Dr. Morris. Thank you, everyone, for coming this morning. I think that was a really nice introduction to what I want to talk a little bit about today, which is the study we're proposing this summer. When my family and friends learn that I work with the Knight ADRC, they always ask me, will we find a cure for Alzheimer's disease? And I always say, I truly believe that it is coming soon. I work with these incredibly gifted people that are working so hard. That is one secret that we have at the Knight ADRC. The other secret is I've worked with you, and I am always so incredibly impressed with the devotion that you have to this research. And so I know we've said it over and over again, and we will continue to say it. It is because of you that this disease will be conquered. So thank you. So let's talk about research volunteering. We've certainly, I guess we're beating a dead horse today, but you will change the course of Alzheimer's disease. You will be the reason that we find a cure. What do you give? We know you give time. We know you haul your family members in. We know you literally give of your blood to us to help us understand what is going on. I know the memory tests are very popular. I hear that. I think you don't realize that how important this information is over time. We pull off those calendar pages, and every year you're in, and it's this longitudinal information that really helps us understand what is happening with this disease and where we might possibly intervene. So sometimes I think this happens. You're sitting there the night before your clinical assessment, and the big snowstorm comes in, and you think, oh, I've been there for the past 5 years. It's really snowing out there. Maybe I'll skip just this one time. I'm just one person. There are 871 people that are coming to the ADRC. Maybe this is my time to take a little break, right? Well, pick up your clickers, if you would, one more time. And answer that question. Click 1 for true and 2 for false. Missing one visit really won't make a difference. Because, right, you guys are devoted. About 50 clickers? How many clickers do we have?
Each table has a clicker.
Okay, Okay, we'll give it a few more seconds. I see about 36 people in. Okay. So one visit will not make a difference. All right, well, let's see. I'm going to tell you a little story, and we'll see if you think that that's true when we're done. Okay, so this is the story of an amazing new drug that was discovered. It treats whatever you want it to treat, something amazing. And these 4 amazing volunteers, Mary, Sam, Samuel, and Danny, have enrolled in this trial, and they are going to be evaluated. They take a test before and after they get this magic treatment. So Mary was, she couldn't make it in that day. She had a flat tire, and she did not make it into her visit that day. She was not able to take the pre-test. And you see, Danny was not able to take the post-test because he was in Florida. He was going there to visit his son. So there was a little bit of missing data, but if you look at the average scores of the before and after, the score before was low, 43, and after the test was 73. So it looks like this treatment actually works. So pick up your clickers again, clicker captains. Let's see, would you take this new amazing treatment for whatever it is that you want to? I know the biostats people I heard that, are killing me over here. I know I'm wrong, Betsy. But let's just imagine that this is a little bit of missing data, and what does it say? We'll give you a few more seconds. Okay, so a lot of you would take, I think I would consider taking this drug, or this treatment, whatever it is. But let's look at what happens if we fill in the blanks and figure out what that missing data was. If we look at Mary's score, actually, she kind of changes the picture, doesn't she? Because her score, her after score, was worse than what her before score would have been. And Danny's score only improved slightly. So when you look at the difference between these people with no missing data, the story changes. And I know this is a bad statistical example, but when you put in 100 people and you look over time and the missing people aren't missing for random reasons, it can really begin to change how we interpret and understand statistics. So even missing one time and the types of analyses that they like to do can make a difference. And so, yes, actually, when you come in, it is important every time. So let Tiffany know, and we will work with you if you need to go to Florida to see your son, or if you have something that goes wrong and your daughter can't bring you because there's a flat tire. We completely understand, and we want to make it possible for you to come in, and we will. Retention is important in these types of studies. For other reasons. We're starting, as you saw the numbers, we're recruiting younger people who may not have been touched by Alzheimer's disease and may not have the same drive and passion for seeking a cure that many of you I know feel. We're recruiting more healthy people who may think that because they have no symptoms, it's less important that they participate. In fact, we know that that is not true, but we know that there are younger folks coming in that might hold that opinion. We understand, John has said it, these studies are complex, and you do really tough things for us, and you come in multiple times, and we are grateful. But those multiple times make it more difficult for you to not have missing data. And we know we ask so much from you. So retention is important, and we do want to make it as easy as possible for you to participate. So you can be sure that if there's a snowstorm, somebody like John Morris will be out shoveling the sidewalk for you to make sure you can make it in. But we want to go a little deeper than just depending on John to shovel for us. We want your help, and we want to know exactly what we can do without overburdening you anymore to make participation easier and better for you. So we want to identify what are the barriers and what are the facilitators that we have within our existing structure that can help you come and not miss any visits for us. So beginning in 2016, June 2016, which is now, actually it's going to start after the 16th, we will be asking your opinion We have some short surveys and we have some open-ended questions, and we want to hear anything. And I will not share any names with anybody, so you can bet that I will keep that secret. Even from my colleagues at the ADRC, they won't know names of people. We might describe you demographically, but you can be sure that we will keep your information confidential. But we want to know honestly what we can do to improve. I want to point out Hamza Ekbal, a student that will be joining us. If you want to stand up, Hamza. Hamza is a volunteer summer student. He will be actually one of the research assistants that might be contacting you either in the waiting room over phone, so thank you. And his a colleague, Pauline Hidalgo, that will also be joining us. So let's vote again, because we love the clickers, and you're getting really good at it now. If you were not able to come into the Knight ADRC for a visit, would you be willing to talk to us over the phone if there was a reason that you couldn't do it? I want to know what I will do to really completely annoy you. And so that's why I'll stop hopefully right before I get to that point. So go ahead and vote. We want to make sure if it's okay if we give you a call. Okay? Great. You guys are the best. This is why we will. Thank you. Another vote. So if we were to develop this survey, there's a couple options we've been thinking of. One was to talk to you in the waiting room before your clinical visit. The other was over the phone, maybe web-based. I don't know how, I'm not very web-savvy, but there are young people that can help me figure this out, or paper. What kind of survey would you actually prefer? In person, number 1, over the phone, 3, web-based, 4, paper. Okay. All right. Will somebody write this down? Does it save it?
No,
so preferences in person.
We got the first two.
Okay, so those are the two methods. The first two, in person or over the phone, are the primary. If you get to the waiting room and you don't want to talk to us, you let us know and we'll call you. Thank you. We'll be talking to you soon.
All right. Did I confess to you that my memory isn't very good? I neglected, when we were introducing faculty, to recognize someone who has been with the Memory and Aging Project as long as I have, and that is Dr. Betsy Grant from our Biostatistics and Data Management Corps. Betsy, we're going to stand up over here. And I also neglected to indicate, when we're looking at signals that Alzheimer's in the brain, that one of our faculty has been interested in whether that can be detected by an eye examination. Dr. Greg Van Stavern is in the back of the room. He's standing up and waving his arm. And if you want information about that, please see him. Then finally, when I was talking about Dr. Lucy's program with sleep, I didn't give you the bottom line. More sleep, less Alzheimer's. Not bad, right? All right. Now we're going to hear from Dr. Bo Ansis. Bo, are you here? Bo's coming up to the podium. So Bo is been with us since 2009? 2008. I told you my memory's not very good. And he originally was interested in looking at people who are HIV positive through imaging techniques, but in recent years has included Alzheimer's disease into his study. So Dr. Ansis.
So can you see me in the back? So really, first of all, I just wanted to say it is a great pleasure to be here. It's a great pleasure to see a number of the participants that I see at the imaging when we do this. And really, again, we would not be here and I would not be able to be presenting some of the results of what your brains look like if it weren't without you. So I really wanted to say thank you, and we really want to show you what we're learning from these kinds of scans. So as John was talking about, we're really been, and most of our studies in the past have really been looking at Alzheimer's disease and really only the tip of the iceberg. And so as you've been hearing, a lot of our treatment studies have really looked at individuals who have symptomatic Alzheimer's disease. And as Dr. Morris was previously presenting, it is really a slowly gradual process. And our hope is to actually uncover more of this iceberg and see more below. And now with some of these new imaging techniques that we're going to be showing you some of these results, I think we're starting to be on that road. And I think there's some very exciting prospects in the future, and it's really because of, again, your participation. So as Dr. Morris was talking about, what you see on this graph is that as individuals with increasing years of age, a number of our individuals with normal aging have very minimal declines in their cognition, and that is normal aging. However, in some individuals, they kind of fall off, and they start to have progressive declines, and they can have what we see as cognitive impairment and then develop dementia. And so what we're now very interested in looking at is that transition, as Dr. Morris was saying. And do we have now new imaging agents that can start to look at that transition? And what I'm going to say to you is that I think we do. I think we're starting to get there. And because of that, I think we're now going to also have potentially new therapies that we can start to evaluate because of these techniques, because of the lumbar puncture, because of these imaging methods as well. And so really, how do we make a diagnosis of Alzheimer's disease? And we also have a neuropathologist, Nigel Cairns, in the audience as well. So I'm helping out John here for a second. But Dr. Cairns,
Have Nigel stand.
and Nigel should stand here for a second. So Nigel is also a world leader neuropathologist. And so a lot of our studies, and really our only way to make a definitive diagnosis of Alzheimer's disease, is to look for these two characteristics. So one is these amyloid plaques, and second, which you've been hearing, is these tau neurofibrillary tangles. And it's really the combination of those two of those that allows us to make this hallmark diagnosis. And so what we're now looking for are agents that can look at these same kinds of things, so can look at amyloid and can look at tau in the brain. And so as John has been telling you before, previously, for a number of years, we've been having amyloid agents. And so a number of you have actually participated in this, and we've been looking and being able to see these early changes in amyloid. More recently, we are now being able to look at tau imaging, and that's what I'm going to be presenting a number of results to you. And so what we've been starting to see, and some work that just has recently come out, is we have been able to look at healthy individuals, so these are healthy or cognitively normal individuals, and looking both at their A-beta as well as their tau images. And so some of the individuals, some of the people in this room today have participated and have had both of these kinds of scans. And what you can start to see is that in our individuals who are cognitively normal, there is some mild amyloid that's deposited. And we know that because we know that these changes occur very slowly. And then even in individuals who are 70 years old who have no impairment, if they go to autopsy, about 30% of them actually have amyloid present in the brain. In contrast, when we look at the tau imaging, we don't really see a large amount of increases in tau. Now, you do see these little red spots in the deep part of the brain. We think that that's actually something we call off-target or just something that's binding something else in the brain. Now, the difference is that if we go down below, in an Alzheimer's patient, what you start to see is that there's more red that is present with his amyloid. But what is striking is that there's an increase in the amount of tau. And so even my mom, who could see this, is that there's a large amount of red, and it's really in the back of the brain, and then it's starting from the back of the brain to start to progress to the front of the brain. And so this tau agent could help us in that kind of transition phase when people are moving from cognitively normal to starting to have mild Alzheimer's disease. And so the question is that, yes, we have this imaging agent, but you've also done those lumbar punctures. And again, thank you for doing that. The question is, does that look similar to what we're seeing in those lumbar punctures and that tau measurements that we get from the lumbar punctures? And work that we've been doing with Ann Fagan, who's standing in the back, we've looked at both tau and A beta in these individuals and compared how does that correlate or how does that associate with these tau imaging measures. And what we see is that it nicely correlates. And in particular, the tau that we see in the spinal fluid nicely correlates with the tau in the brain. And so that is that as tau increases in your spinal fluid, we see tau levels increase in the brain. What's very interesting in this is that we can also kind of see where it is in the brain. And so really the tau imaging kinds of helps us kind of say, where are those kinds of changes that we're seeing? So it's a very important measure. It's not only what is occurring, but where it's occurring. And then what we've also seen is that even in cognitively normal individuals who are starting to have changes that are having A beta, so that means that amyloid is starting to change in their spinal fluid, we're already starting to see some tau changes in other areas of the brain. So what this is starting to tell us, and what is very, very important, is that now we're starting to have a better understanding of what's occurring. It's not just probably the amyloid, it's probably the combination of the amyloid and the tau, and that's what Dr. Morris was showing you in those previous pictures. And that's kind of tipping over, and that's allowing for this kind of transition in these individuals. And so it's really the combination of the two. And only by getting both these imaging and spinal fluid are we really able to understand kind of this trajectory. And so really, it's due to you that we're starting to understand this kind of transition phase of individuals. And then you also do those pen and paper tests. And I know, as we've heard before, they're very difficult. And yes, I haven't done well in all of them as well, too, so I know what you're going through. But the point is that are those pen and paper tests important for us? And the answer is those pen and paper tests, as those starting to see changes in those pen and paper tests, they more correlate with the tau imaging measures than the amyloid imaging measures. And that kind of makes sense, is that we see in these kinds of trajectories that you saw before, it's probably amyloid changes, then it's probably neurons or brain cells not firing well, then the tau changes, then those kind of thinking problems, and then the kind of clinical presentation of individuals. So it's kind of, we understand that these kinds of measures are now, with the tau, are much more closely correlating with our performance on those neuropsych. And how we know that is that if you see each of these numbers, higher numbers are stronger correlations of that tau, 0.51 versus 0.0, 0.7 versus 0.12, 0.66 versus 0.10. In each case, these tau changes in the imaging seem to correlate better with those neuropsych performance. So this is a very heady time right now. This is a great time, and we're starting to now learn and understand that. But it's also very important to actually also understand normal aging. And as you've been hearing from Dr. Morris, we have individuals from the ages of 23 to all the way of 101 years of age. What happens with just normal aging? Because if we understand the difference between normal aging and Alzheimer's disease, that will also be very important in our diagnosis. So I did want to tell you about, there are studies that are going on, and some of you have actually participated in them, and a new study is actually going to be occurring. And some of you may have seen this in the news. It's called the Human Connectome Project, and WashU is actually the leader of this project. It's a couple institutions that are doing this, and they are really mapping out with fancy-dancy, and you see all these kind of pretty pictures that you often see, pretty pictures of the brain, but it's really very, very, very advanced neuroimaging that is actually being done. And what's very interesting is now we're able to actually look at these both structure, so how does the brain and its organization look, as well as function, how do two areas kind of talk with each other using these kinds of imaging techniques. And why it's going to be important is that some of those same imaging techniques are things that we are thinking about and actually doing as well at the Knight ADRC. And so we have a number of studies that are going to be involving looking at normal aging. In fact, one of these pictures, in fact, one of our participants, I've been told, was actually on the cover of that famous journal Science, and actually made that because of the work that's being done here. And so there is a new study that's going to be coming through. It's going to be called the Human Connectome Project and the Aging Study. And those are looking at individuals that are 40 to 90 years of age who are cognitively normal. And they'll just be having 2 MRI scans and being followed up. If you want to know, there's not a sign up there. You can talk to me or others about this in the future. But more importantly, I want to, again, at the end, again, stress to say thank you, because we would not be able to have these kinds of studies without you. But it's also, I'm just one of many here. I know Dr. Benzinger couldn't be here today, but it's really a pleasure to work with her. And you see the rest of our team that we have assembled here, and many of them who are here today showing their support. And again, thank you so much for coming out today.
After the conclusion, there will be tables in the foyer that to talk about Dr. Van Stavern's ocular biomarker study, also the sleep study of Dr. Lucy, and the Memory and Aging Project. So I mentioned earlier we have an African American Advisory Board. We're very interested in the Memory and Aging Project, in being welcoming to all individuals. If we only study in Alzheimer's disease in people of European descent, we'll only learn about Alzheimer's disease in people of European descent. And we know that there are differences among groups of individuals, European Americans, African Americans. Are those differences important for Alzheimer's disease? You may hear that Alzheimer's disease, and Dr. Lenice Cummings-Vaughan, who is going to emphasize this, may be greater in African Americans than it is in Caucasians. So we're very interested in studying Alzheimer's disease in African Americans, and we're fortunate to have Dr. Lenice Cummings-Vaughan to talk about our diversity program. Let's welcome her.
I just move it around. All right. Well, thank you again for having me come and speak with you about this. So as my talk says, I'm going to talk about diverse populations. And yes. Oh, can you hear me? I'm sorry. So I'm going to speak about diverse populations. So what do I mean by diverse population? Sometimes people say, is that a code word for something? And I'm not really saying it's not a code word for anything. But when we look, if you look in the news, if you read a newspaper, you already know that the country and the world is changing. There's migration in different places. So we have a changing demographic of not just younger populations, but older populations. So as it says here, I work in the Division of Geriatrics, and I'm especially interested in people who are older. And so in the geriatrics world, we say that we have an aging tsunami. We call it the graying tsunami. And so what is that intersection, or the importance of the changing demographics, both racial and ethnic changes, as well as the graying tsunami? So that's what I'm going to talk about. So first and foremost, the greatest risk factor for Alzheimer's dementia that we know of as of right now is age. And so as we all age, we know that we're all going to be at greater risk. But the significance of this is that, as I said, we have an aging tsunami. So that means we have growing numbers of individuals who are going to be at risk for Alzheimer's dementia. And then if we look at kind of the changing demographics in terms of ethnicity, what does that mean? And so if you look at, I mean, this is pretty significant to think that the number of individuals who are older, and the United Nations, usually they are referring to people who are over the age of 65, that those numbers might have tripled. And we blame baby boomers for lots of things. Can we just blame them for this also? However, we know that this is going to continue. So even after the baby boomers are well past into this aging population, we know that the projections show that individuals who are younger, we call this the millennium generation, that they also have a higher, they are going to age and become even older than we are. So that means if we don't combat this problem of Alzheimer's dementia, we are going to have a great problem, an ongoing problem. So as I said, it's not just aging, it's that the demographics of the aging population as well as younger population in the country is changing. And so we know that, for instance, that by 2060, approximately 1 in 3 individuals will be of Hispanic origin. We know that the percentage of those identifying as Asians will rise from 8% to 10%. And African Americans have one of the smaller changes in percentage of the population, but it's still significant. So we can see that we really have to have our eyes wide open, as Dr. Morris already said. If we are just looking at one part of the population in terms of Alzheimer's dementia, we won't have a true idea of Alzheimer's dementia disease process and treatment for everybody. So if I were to just give you general statements about what we know about differences in disease presentation, as well as treatment, overall in all diseases as well as dementia, we know that there are differences. And acknowledging differences is not a bad thing. I think we have to acknowledge differences in order to combat them, and so that everyone reaps the best benefit from any treatment that we could possibly give, especially for dementia, but for any disease process. So we know that there is a difference in the incidence, so new cases of Alzheimer's dementia, as well as the length of time a person lives with dementia. There is a great difference between those of African American descent versus those who are non-Hispanic whites. Why is that? We need to ask ourselves what the reason is. We know that in terms of participation in trials, not just in dementia trials, but any medical trial, that there are significant differences among different races and ethnicities. The use of medications can be different. Use of services, so home health or use of nursing, these can be different. There can be cultural differences in ability or desire to access these services, whether or not it is a family expenditure or it's an expenditure that's mostly footed by insurance, that there are racial and ethnic differences there. The quality of care we know has been different for different medical conditions, and that caregiving can also be a difference. So how I think it's important to take care of my grandmother might be different than how another person perceives that. So we do have studies that show already what differences or similarities there are. So we know that African Americans are twice as likely as non-Hispanic whites to have any dementia, and that 50% more Hispanics have dementia. So that's pretty significant, and we need to figure out why that is. We know that the prevalence, so that means a person just living with the disease, not a new case of the disease, that prevalence is higher in those of African descent. And is that just because they are diagnosed earlier? Is there another reason that a person who is of African descent is living longer with the disease? We also know that there might be differences in the expression of genes that place people at higher risk of having Alzheimer's dementia, that there are differences in the expression of those genes. And when we think, is there a difference in expression, does that mean that that's the reason that there's a difference in Alzheimer's dementia prevalence? Or is it the interaction? We've already heard about the biomarkers. Is there some interaction between lifestyle factors, these genes, some interaction that causes this increased risk? So Alzheimer's dementia doesn't exist in isolation, and we all have other medical conditions that we're at risk for. And we know that African Americans have a higher rate of hypertension, of obesity, as well as diabetes, and these are risk factors for Alzheimer's dementia. Is that interaction between these disease processes causing a higher risk? As well as other chronic conditions like depression or chronic stress that might be a factor in other ethnicities, as well as in the non-Hispanic white population or Hispanic population. Is this something that we should be looking at? Lastly, we already spoke about these diagnostic tests that everyone loves to take. And, the truth is most of these tests were developed in populations that are not the only population that it's being tested in. And is that significant? It could not be at all. We don't really know. And, the prior speaker talked about how changes in the tests are seen in the changes of the tau in the imaging. Would that still be true when we look at different races? We don't really know that unless we test that. These are just some more information in terms of racial and ethnic differences. So Alzheimer's dementia is not the only type of dementia, and there are plenty of people who have vascular dementia or have mixed dementias. We know that Asians are at higher risk of having vascular dementia, but we don't exactly know the reason why. And when we look at Hispanic population, we want to see what are the reasons why there might be a higher risk of dementia. Is it because of the higher interaction between other disease processes like cerebrovascular disease or diabetes that place people at higher risk? Is it the expression of these genes that we know that place others at higher risk? We don't know the answer to this. So let's talk about what's available because of everybody that has participated in these studies. So again, this is kind of repetition, but I am so thankful that everybody gives of their time to participate in this study. And the Knight ADRC has done an incredible job of recruiting and retaining participants. And it's because of that I think that we're able to ask some of these really compelling questions that are important overall, not just here in St. Louis, for the study of Alzheimer's dementia. So because we have a diverse population to test, we know that we can look at some differences in the biomarkers. So we have enough cerebral spinal fluid samples, imaging data, that we can have these hypotheses. So is it possible that the reduced CSF concentrations in amyloid beta, that they are the same between both populations, and therefore that's not potentially the reason for the difference? The elevated CSF concentrations of tau and phosphorylated tau, that those are also the same, and that there might be an increased retention of A beta tracer detected, that potentially those are all the same. So that's our hypothesis, that although we know that there are differences, we believe that some of these things, the biomarkers, might all be the same. And then we have to look at what are the real reasons that there are differences. So you can see, and again, a testament, 80% to 20%, which is really representative of the population at large in the St. Louis area. And I think that's a testament to the great retention and recruitment of this Knight ADRC. Some other questions that we might ask, and I think that I'm open to more questions. So I think in our question and answer session, maybe you all can come up with some additional questions we might ask based on the possibility of further analyzing the data that's here. All right. So last, I want to show you a sample of what we have. So these are participants in the adult child study, and this is the level of completion of lumbar punctures, PET scans, and MRIs. And you can see that although that there are differences, we do have significant numbers in both groups that are completing this. But I think just as all the prior speakers have said, the more that everybody participates and fully completes all of the testing, the more data that we have to really analyze and see if there are differences and find out what the interactions that cause Alzheimer's disease presentation and how we can treat Alzheimer's dementia. So I'm not going to harp on, that 79% are getting an MRI. I think it's wonderful that 79% are getting an MRI, but I think it's really important that we all want to help you to fully complete all of the testing. Thank you.
Thanks, Lenny. So the last presenter will talk about these clinical trials that I mentioned earlier, and that's Dr. Joy Snyder. So will you please welcome Dr. Snyder?
Good morning. Are your brains full yet? So I won't take too much more of your time because you already heard a lot, and some of what I'm going to say you've already heard, but I just wanted to give you an update on where we are with treatment for Alzheimer's disease. And let's see, start with that. The first thing is to remind you, and I'm probably preaching to the choir here, but Alzheimer's disease is a serious disease. It's one that affects a lot of us, and it's one we haven't made as much headway on recently as we would like. So the picture you see up here, and there's that red dot, shows you a number of different diseases, which I can't even read on my monitor, but a number of different diseases. And over the last several years, we've made a lot of progress in many of these diseases, in cancer, in heart disease, and stroke. The number of people who are dying of these diseases has actually gone down, which is great. The one pointing upward is Alzheimer's disease, and the number of deaths from Alzheimer's disease has gone up by 71%, which is a lot. A lot of this is driven by what you were just hearing about, the demographics, the aging population. More people are getting Alzheimer's, more people are dying of it. But we want to make some headway here, and we want to begin to make this line go down as well as the rest of them. And that's what this is all about, and that's why we're all studying Alzheimer's disease. 1 in 3 people will get Alzheimer's disease when they're older, and they'll die with Alzheimer's disease. And that may be an underreporting because it's often not listed on the death certificate. More than 5 million people right now have Alzheimer's, and that number is going to go up. So that's a lot of folks. And as Dr. Cummings-Vaughan mentioned, it is more common in African Americans. So I think are 2 times more likely to have Alzheimer's disease. So a very common disease. So we all are interested in making it better. Okay, great. So you've heard a lot of this before, but I just want to lay the groundwork for the clinical trials. And the first thing is to define what we mean by Alzheimer's disease, and that seems pretty simple. We've been studying it for 30 plus years, but in fact our idea of what the disease is has changed over the last few years, in large part due to studies done here, and Dr. Morris has led the charge here. And the rest of the world is coming along to our point of view, in large part thanks to the data we have from you. But by Alzheimer's disease, we're talking about the brain disease, the changes in the brain, those plaques and tangles you've seen on the slides already today. That's Alzheimer's disease. We used to think that meant somebody who was severely demented, but now we know there's really 2 stages to Alzheimer's disease, and it's like that iceberg Dr. Ensis showed you. The stage we thought we knew, the one that's above the water, is really just the tip of the iceberg. Most of Alzheimer's disease is in that preclinical stage, people without any symptoms at all. So really, the symptoms that we see are just a tiny part of it, and it really is mostly a preclinical disease. And then people develop symptoms, and the symptoms span the gamut from very mild, where we kind of scratch our heads and say, is this really a problem, to more obvious, where we say, yes, they have dementia, to severe dementia. The other thing that our center has sort of taken the lead in is Alzheimer's disease is not a change of you from the average person. We give you all those psychometric tests, and we can compare them all to averages with various norms. But really what you want to know, and what we want to know, is how have you changed yourself? So intra-individual decline, where you are from your baseline. And we want to know that it interferes with daily function before we say it's symptomatic. So you've seen this graph in a number of iterations, but this is the timeline of biomarkers. And this is still, in many ways, a hypothesis. We know much of it is true, but we can't nail down the specifics. But this is the hypothesis of how the biomarkers change over time. And I was very jealous because Dr. Morris's slide is way slicker than mine. But bear with me. This is the one he let me use. So, but the first thing we think that changes is that cerebrospinal fluid, that fluid we get from those lumbar punctures that you all love to have. That's the very first thing that changes. The levels of that amyloid beta peptide in that fluid go down. The next kind of changes, we see that amyloid imaging change. We'll see changes in the tau in the spinal fluid. Then we begin to see changes in your brain imaging. And then much later, so out here in the graph, we begin to see changes in your memory and thinking that we can detect. And the key point is that there is this 15 to 20 year time difference between when those first changes are in the spinal fluid and when we begin to detect those changes in your memory and thinking. So that's that preclinical period. And that's something 10, 15 years ago we knew nothing about. So that's really the big change recently in the field. So what about this amyloid imaging? You've seen some pictures of that, and this is amyloid imaging with this Pittsburgh compound B. And what you can see is the normal person doesn't have a lot of red stuff, and the person with Alzheimer's does. So those are those amyloid plaques in the brain. That's that half of that autopsy we're going to be able to do before you're deceased, which is great. This is a person who's normal, and guess what? These two look exactly the same. So they look exactly the same. This is that preclinical Alzheimer's disease, and in this case, 3 years later, this person develops symptoms. So we know that's what preclinical Alzheimer's disease looks like. So the first study I want to tell you about, now that we've gotten all the background out of the way, is a study called the IDEAS study. And I can't tell you what the acronym stands for. I think there are committees in dark rooms who come up with these things. But the IDEAS study is actually a very interesting study. So the amyloid PET scanning agent called Amyvid or florbetapir was approved 3 years ago, and there are two other ones very similar that were also approved. So you can go to the radiologist right now and get one of these amyloid PET scans if you have $4,000 handy that you would like to spend on it. So the issue with these studies, and the reason we don't use them in the clinic that often, is because Medicare would not reimburse for them, even though they were FDA approved. So the idea was that maybe we should reimburse for these, but we want to know if they make a difference in clinical care right now. So the IDEAS study is a study to use these scans in people who come to the doctor with a memory complaint. And so these are people who, if the scan is positive, it means they have amyloid changes in their brain. They're going to fund about 10,000 people over the next few years to get these scans, again, through your doctor. It's not through memory and aging. This is through when you go to see your doctor if you have a memory problem. And these folks can get the scan if they're over 65, eligible for Medicare. More about that on the next one. So this is their slick brochure you can find online, or we can give you at the clinic. And it's going to help us learn whether right now in clinical practice, whether this amyloid scan will be helpful for diagnosis, for treatment, whether it'll be helpful for patients and families. And this is a great thing, and our clinic patients are very excited about it. We don't know if it will be helpful because right now we don't have amyloid-specific treatment, so it's still a very good question. The key thing for you to know is the only reason we could do this study, the only reason this scan is available, is because for the last 10, 15, 20 years, you've been having amyloid PET scans and spinal fluid markers and cognitive testing in studies like our ADRC, and we are certainly one of the leaders in this field, and you help make this possible. What about prevention studies? Dr. Morris mentioned this. So this is the really exciting thing happening in Alzheimer's disease, is that we're now that we know about this preclinical period, maybe we could treat people. Maybe we could prevent the disease. That would be great. We could all live our lives as long as possible with our memories intact, which is what everybody wants to do. So the first study to look at this in people that don't have a gene mutation is this study called the A4 study. And again, some committee came up with this. I had to write it down because I can't remember. It's anti-amyloid treatment in asymptomatic Alzheimer's. So that's 4 A's. I have no idea what the extra A is in A5. I guess we'll find out. And I heard A6 may be in the planning table, so who knows. But the goal of this study is to prevent memory loss in people who have a positive amyloid scan before they develop symptoms, so people who are perfectly normal. They're going to be treating with an investigational treatment. It's an antibody called solanezumab. It's been tested in people with symptomatic Alzheimer's disease. We don't know as yet if it works there, but we're testing it in people that are completely normal. People will come into this study. They'll get this Amyvid scan to look for amyloid using this compound florbetapir, and a whole lot of people are going to have to be enrolled because we don't know when exactly these folks would develop Alzheimer's. So 500 people will get treatment, 550 will get placebo. That's pretty exciting stuff. People have to be 65 to 85. They have to be normal, no memory problems. You have to have a study partner. and they have to be willing to participate in a lot of procedures, including the MRI, the Amyvid scan, the LP. They're going to get treated for 3.5 years. It has to be a really long study because we don't know when these folks would develop Alzheimer's, and probably the majority of them will not, but this was calculated so enough of them would, we could detect a signal. Again, like we mentioned a few minutes ago with Dr. Cummings-Baum, the focus is on diversity. We want to know if this works in all populations. We want to find people over 75, not just 65, people who have a family history, people who don't, people from various ethnic groups. So that's pretty exciting. The study is now at about the halfway mark nationwide, which is exciting. WashU was slated to enroll, I think, 10 people, and we're 15 people, and we, I think, are on about our last person, so we're almost done, which is great. So that's pretty exciting. It's been a very challenging trial because you've got to screen a lot of people to find folks with positive scans, but it's been terrific. And don't know if our study coordinators are here for that today, but they've been working incredibly hard. That's Erin Couture. She's around, so thank Erin for that. What about people, so A5 is coming, so another study is coming. So A4, we're almost through recruiting for, but don't worry, there's more to come. So you'll hear more about that. There's always more to come until we get this disease stamped out. What about people who have symptoms? So, we're all excited about this preclinical stuff, but that's great. But what about the folks who already have disease? We're not going to ignore them. We certainly want to treat them. So as Dr. Morris mentioned, there are a couple of studies coming with that. One is a study, and all these drugs have great names. I hope you notice that you really can't say. But a study, I call it aducanumab. I'm not sure what anybody else calls it. This is called the EMERGE study. And it's, again, another antibody that may help remove Alzheimer's plaques. The goal is for over 1,300 participants worldwide, and we want to have 10 here at WashU. We haven't started this study yet. We're just working up to it. But this is a pretty exciting study because it, again, targets people with very mild Alzheimer's, but people who do have symptoms. They're going to get one of these scans, an amyloid scan, and only people with a positive scan go into the study. And this is sort of the trend now. We want people that we know have Alzheimer's pathology to be in these studies. And they'll either get active drug or placebo. What's interesting about this study is people will be treated for 18 months on active drug or placebo, and then they're going to have an open label where everybody gets the drug. So that's pretty exciting. So we're excited about starting that study. The other study that Dr. Morris mentioned is the study of what's called a BACE inhibitor. It's a pill that you take that will keep your body from making as much of that amyloid protein. That is due to start here at WashU pretty soon. And that study is one, and it's got a great name, LY32, whatever. We just call it the BACE inhibitor, and we go from there. It's an oral agent. They're looking for 1,000 patients worldwide, aged 55 to 85, who have mild Alzheimer's disease. And again, that amyloid PET scan will be given, and if you don't have a positive scan, you don't get enrolled in the study. So we only want to treat people that have, absolutely for sure, have Alzheimer's disease. It's treatment for a year, and the main outcome measure is going to be the tau imaging. So you have to have the amyloid pathology to get in, and they're going to use the tau imaging to see if the treatment is working. So that's very exciting. And again, based on the imaging studies done here and elsewhere. What about people with Alzheimer's that runs in the family? And this is that very rare form that attacks people when they're younger. So as Dr. Morris mentioned, the dominantly inherited Alzheimer's network, or DIAN, this is the treatment arm. And this is a very rare disorder. We do have a lot of participants here in WashU, but they fly in from all over the country. So it's quite a rare disorder. They must have a mutation that causes Alzheimer's that's been tested for, and we know it's there in their family. The folks that are pre-symptomatic, which are most of those in this study, have to be within 10 years of when we think they're going to get memory problems. And they, again, receive one of these antibodies as part of the study. It's an international study. There are 210 participants worldwide, and the study just closed to recruitment a couple of months ago, which was very exciting. This was a study when Dr. Bateman first proposed to do it. Everybody told him it was impossible. But as usual, he went ahead and did it. So that was good. At any rate, that's an exciting study. It did start at WashU. It was the first prevention study. And even though what we study here with most of you is the sporadic form of the disease, the groundwork laid here made that study possible. So you should be proud of that as well. So what's going to happen in the future? So if you go online, if you want to know what's going on, and you go online, you go to clinicaltrials.gov, and you look at how many Alzheimer's trials there are, and there are a lot. There's about 200 active intervention trials with something, a drug, some sort of manipulation, dancing, whatever, for Alzheimer's disease right now in the United States. There's many new studies planned. We talked about A5. We talked about, actually, the DIAN-TU will be adding a new drug arm in the near future. The emphasis in all of these studies is on biomarkers, on the imaging, on the spinal fluid, to make sure that people have the disease, and then to track how well those drugs actually attack the disease. So that's really exciting. This allows us, again, for the first time, to take people who are normal and try to prevent Alzheimer's, and allows us to confirm that those we're treating do have symptomatic Alzheimer's. The bottom line is that we've been working at this a long time, and I used to show a slide of all the trials that had failed, and I quit because it made me depressed. And it's hard to depress me, but now we have good reason for optimism. A lot of these trials coming online are new medications. We're learning when to use them, which may be really important and something we did not suspect in the past. And these new drugs are attacking the underlying brain disease, which is very exciting. They're still mostly amyloid treatment, a lot of amyloid antibodies and things that affect amyloid, but tau treatments are coming, and there's some studies now in phase 2 and 3 looking at tau treatments. So we're pretty excited. Again, just pat yourselves on the back if you can without hurting yourself, because the biomarker studies that you did, all those spinal taps and imaging and cognitive testing that you've had done, and clinical assessments for sure, really made all these things possible. So thank you for making this possible. I think it's a hopeful time in Alzheimer's, in large part thanks to you. And that's all I have, so I guess we'll take questions. Thanks.
Thanks, Joy. So let me invite Dr. Bo Ansis and Dr. Lenice Cummings-Vaughn to join Dr. Snyder here at the table for a panel discussion. And all of you can fill out your questions on the index cards that are available to you, and hold them up, and we'll have our staff come around and pick them up and bring them to the podium. Now, as the cards are coming up, I had 2 questions posed to me this week by one of the participants. The question was, we asked a participant to give a blood sample at one of her follow-up visits. And this has been a change. We usually got blood only at the very first assessment, so she wanted to know why we've changed. Why are we getting blood at follow-up assessments now? And we failed to really be able to explain that to all of you, so I'm going to explain it now. We have great need for blood. That's where we examine the DNA that gives us the genetic information about risk for Alzheimer's disease. Dr. Lenice Cummings-Vaughn talked about that a little while ago. And then the second question was, we look at other things besides DNA in the blood. We look at something called hemoglobin A1C. Have any of you heard of that test, A1C? It's for diabetes. And the participant said, my doctor gets that. Why do you have to get that? And the reason is, in research, we have to do everything the same way with the same methods in the same lab. So your physician's A1C will be done in a different lab than our laboratory, and we can't use it because we don't know how well that compares. So we apologize for not making those explanations for you. Okay, so a number of questions here. I'll try to get them organized. Okay, Dr. Ansis, what is an amyloid scan? You heard a lot about that, and how does that differ from a PET scan?
Okay, thank you. That was a good question. So if we just think about this for a second, the PET scans are the major kind of scan that we do. There's 2 ways so PET stands for positron emission tomography. And then there are 2 major markers that we're looking for. So one is for amyloid, and then one is for tau, okay? And then of the scans that we see, or that for amyloid, one is, you've heard in the past, is something called Pittsburgh B compound, or PIB compound. And then more recently, a newer compound has been developed that's an amyloid scan that also allows us to look at amyloid in the brain. And then for the tau imaging, there are a number of different names and that are being used. Sometimes it's called AV1451 or T807, so all these numbers. And then there are a couple other newer compounds that are also being used. But that's the two main parts are amyloid and tau, and they're both types of PET imaging.
So an amyloid scan is an amyloid PET scan, whether it's Amyvid or PIB or whatever it is.
Right. So an amyloid is an amyloid PET scan. They're all use the PET imaging method. So that, again, is where we inject the tracer and then wait a period of time, and then we see how much of an increase or uptake is in the brain. And then, as you saw in a number of the different slides, then we can see if there's an increase, and we look at that, and then we can make it those pretty colors. But really is what we're looking at is increases in amyloid deposition, or if a PET scan for looking at tau, we're looking at increases in tau deposition in the brain.
Thank you. Lanice, do statin drugs make your memory worse?
I know that there are, I guess there have been some reports about the impact of statins on memory and thinking. And I think there's variable penetration into the brain across the blood-brain barrier of different statin drugs. So as of right now, we often will recommend the statin that has less penetration, and thinking that if it's possible that statins do cause any cognitive changes, that would decrease the risk. But do understand that we do think that vascular changes that are caused by high cholesterol are a risk factor for vascular dementia as well as Alzheimer's dementia. So not treating the high cholesterol is also a problem.
Thank you. Joy. The world lost an icon yesterday. Muhammad Ali passed away. What is the relationship between head injury and brain disease?
Getting hit in the head is bad. So that's the bottom line. So there are forms of dementia that have been linked to brain injury. And I believe Dr. Ali had a Parkinson's disease related to brain injury. That's linked to repetitive brain injury, so that's called dementia pugilistica is one term for that. There's also, everybody's heard about all the NFL players who now have something called chronic traumatic encephalopathy, and that can certainly be linked to brain injury and getting repetitively hit in the head. So those things are clearly linked to trauma. The links between Alzheimer's and trauma are a little less certain, but certainly if you get hit in the head a lot, in most studies, you would be a little bit more likely to have Alzheimer's. So the bottom line is to take good care of your brain and don't get it banged up. And if you do things that might injure your brain, like riding a bicycle or playing a sport or, I guess, boxing, please wear proper head protection. And if you get knocked out, stop doing it. So take care of your brain.
It's a very interesting, very complicated, but potentially very informative area. The first report about head injury and dementia was in 1928, a study of boxers. They called them punch drunk syndrome. They later changed it to dementia pugilistica. And now it's called chronic traumatic encephalopathy. You've heard of that, some of you, through the NFL, right? The NFL? That's not Alzheimer's disease because it doesn't have amyloid, but it does have that tau protein. So there is a connection. What Dr. Snyder was saying, if you have head injury, years later, are you at higher risk of getting true Alzheimer's disease? And the jury is out, but a lot of information suggests yes. We don't know for certain. A couple of questions. Is the United States the leading country for Alzheimer research? Yes. It invests more federal dollars into research than any other country, and it has a network of federally funded Alzheimer's disease research centers that other countries do not. That's not to say that there isn't terrific research in Alzheimer's disease going on in other countries. it is, there is great research going on, but it's not as well coordinated or as highly invested. Next question is, so we're fortunate. So far, we've been getting our grants renewed and totaling up $50 million. That's an amazing amount, $50 million. It's hard for me to comprehend that I'm rich. But I'll tell you something, it's not enough. Dr. Snyder showed of all the major killers in the United States, Alzheimer's disease is the only one for which deaths are increasing because we have no effective therapies, unlike cancer, unlike heart disease, or preventions. So we need more investment. And the question is, are you pursuing additional funding? We are pursuing additional funding. The Alzheimer Association, the National Alzheimer Association, as well as our St. Louis chapter have been terrific partners with us for many aspects, but they've also funded us, and they particularly have funded the DIAN study. We have other foundations that provide us with grant funding, and we have philanthropy. And we know that there are many ways that people can give. You can give of your time, your talent, and your treasure, and we appreciate all of those. We're very much partners at Washington University School of Medicine with the Barnes Jewish Hospital and the Barnes Jewish Hospital Foundation. I'm going to introduce Tina Hissong from the foundation at Barnes Jewish Hospital, who has helped bring in some funding for Alzheimer research at the Knight ADRC. Tina, will you stand up? Thank you. All right. So, Dr. Snyder, did you say that 1 in 3 older adults are going to die with Alzheimer's disease? Is that true?
I said that, yes. That comes from the Alzheimer's Association fact sheet, so I hope it's true. It's a very common disease in older adults, and we talked about as we get older, there are more folks with Alzheimer's, and the current estimate is that if you're over 85, you have about a 50% chance of having Alzheimer's, and as more and more of us age, more and more people will be dying with Alzheimer's disease.
Yeah, so another question had to do, is there something unique about United States, or is this a statistic that goes across the world? And I would say there are no clear geographic factors in Alzheimer's disease. The number one factor is whether the population is going to age. All developed countries, Japan has the most people, percentage-wise, of Alzheimer's disease in the world because they're the oldest. Okay? So it's a matter of aging. Lanice, I'm going to give you a tough one. Are the pills advertised as memory-enhancing over-the-counter drugs of any value?
I don't know which pills those are, but as long as I'm just going to say that, as everyone has already said, we don't have a treatment. So we don't have a treatment. That means no.
What about coconut oil?
No.
What about coffee?
Keeps you awake.
All right, Dr. Ansis. This is not an imaging question, but what's this idea that infection might have a relationship to amyloid plaques? What's going on about that?
Yeah, so there's been, you may have seen in the news more recently, there was some ideas that were proposed that amyloid may actually be somewhat protective and in fighting off some infections. And so one of the ideas that's been proposed is that when there's an infection, the amyloid actually goes and is fighting off that infection, and then these plaques are developing. That was only done in a test tube. It has not been really looked at in a more sophisticated way. Most of the analyses so far have not linked a particular infection to Alzheimer's disease. But again, further studies need to be looking at that. However, there are some studies, and there are some studies that have been looking at that, of is there, with amyloid and other toxic species that are made with Alzheimer's or occur with Alzheimer's disease, is there inflammation that goes on with this? And the answer is probably yes. And then there have been some large studies that have looked at that have been kind of equivocal on looking at the role of inflammatory or reducing that inflammation with Alzheimer's disease. But further studies need to be done in this particular area, and I think that's one of the areas that is gaining increased investigation of looking at the role of inflammation with Alzheimer's disease.
Dr. Ansys, what's this human connectome project, and are there exclusion factors? Someone has asthma and they get weekly injections. Are they able to participate?
So again, the Human Connectome Project is a large study that is at a number of different institutions, and that's to look at normal or healthy aging, and that's from ages from about 40 to about ages of 90. There will not be as many of the cognitive tests that are being done as being done through the ADRC, but it is linked with a number of our participants, so it will be available to individuals like yourselves to do some of this more advanced, those pretty pictures that we were seeing before. There are not a lot of exclusionary criteria. If you can't lie flat in a scanner, then of course, then that would be an issue. Of course, if you have metal or a pacemaker, that would be an issue.
So titanium?
Most of them, the answer is no. We are not allowed to do that. Sometimes people say that they are approved. We need to know ahead of time. We need to have those little documents. So if you've seen your doctor and they've put a piece of special hardware, please ask them to come in with, please ask them for that card so that you know. That can make our lives a lot easier.
So if someone had questions about the A4 study, the Connectome project, where would they get that information?
Come to me, or we will be starting to put that out. That's coming very much in the near future, so the next couple of months.
But if people have questions, they can come up and talk with you.
Just talk to me.
Look at this. You guys are terrific. Somebody pre-populated their question card. They typed it out for me. They came prepared. All right. So, Lanice, I'm going to bounce this one to you since you did so well with the statins. What about other drugs that affect mental ability? So, example, what about the protein pump inhibitors? What about benzodiazepines like Xanax that people use for sleep, antihistamines? Are those bad for you, or do they cause Alzheimer's?
I wouldn't say that any of them necessarily cause Alzheimer's, but when we evaluate patients, say in my geriatric clinic, we always are looking to decrease the number of medications that might affect a person adversely. So yeah, all of those medications have potential adverse side effects or long-term consequences. So if they are no longer necessary, then it's best not to take them.
So they can dull the mind, right?
Yes, they can slow. Well, the benzodiazepines like Ativan, they can make you a little bit loopy. For sure. The proton pump inhibitors like Protonix and things like that, they don't do that, but they have other consequences. You take them for a really long time, you might end up, you have a higher risk of having osteoporosis. So if you don't need it, then we discontinue it.
Right.
Yeah.
Okay, another question had to, someone mentioned about exclusion criteria with asthma, somebody else about titanium, somebody else about a heart pacemaker. Bo, is it the case that it depends on what pacemaker it is, whether someone can enroll and get the imaging studies?
We've been trying to reduce, so most of the pacemakers, even though they may be, quote, MRI compatible, that may not always be the case. So the answer is trying to reduce, or if those individuals do have a pacemaker and it's documented and that, we try to say no, that they shouldn't probably be doing the studies. We do ask you to bring in, again, those cards. So if you do have a pacemaker card and that kind of stuff, or you have other kinds of equipment within you, please bring those kinds of cards, because that can help us and figure out what is or is not MRI compatible. But usually the answer is, with a pacemaker, any kind of pacemaker, the answer is no.
Not eligible for MRI because that's a magnet.
That's a
and
magnet,
it
so
could
they're
change,
not
change
yes.
the pacemaker.
Well, we'd actually not just change it. It would be taken out of them because it's such a strong magnet, it would be taken out. So that's why we don't do. So the answer is, or heat up is what it really does. But typically what the answer is that if they do that, they actually try to turn off the pacemaker. But again, the reason why the person has the pacemaker is because they have a medical problem that they need it. So we say, we thank you immensely for wanting to participate, but for the MRI and for your own, and again, the top issue for us is your safety, we would say, no, please do not do the MRI.
Joy, you gave a very thorough review of clinical trials that the Alzheimer's Disease Research Center at Washington University is going to launch or has already launched. You mentioned there are many more trials of many other agents, not necessarily being done at WashU. But boy, there was a lot of talk here today about tau, and I don't think I heard you say any treatment trial that targets tau. Why is that?
So it's a great question, number one. And there are treatment trials coming down the pike that do target tau. We would certainly love to be involved in them as time goes on, I think would be great. But at this point, none of them were at the stage where we felt like we could participate. Some of them have been very exciting. Several years ago, there was a tau agent that was all the talk at the International Alzheimer's meeting. That one was called REMBER. Again, that committee came up with a great name, I guess. But now nobody talks about it anymore. So apparently the phase 2 trial didn't go well. So unfortunately, a lot of things get started in trials and don't make it to the last stage. Less than 10% of drugs that are started in the testing pipeline end up at the end. So a number of tau agents are out there and are being tested and are coming down the pike. But right now, there are none that we're participating in. There are a number of studies going on nationwide. So we're very hopeful because we do need to target amyloid and tau and inflammation and anything else we can find a target to stop this disease.
So Dr. Ansis gave a number of slides showing the tau PET scan and how you can look at tau through PET, amount of tau, and the distribution of tau in the brain. So why the heck do we still need to do a lumbar puncture to measure tau? Why do we need to do that? So the answer is the tau change in the spinal fluid happens 5, maybe 10 years before the tau PET scan becomes positive. So if we want to identify the earliest change, it's the spinal fluid. Okay? So, yep.
The other thing is that we're still in the infancy of tau. So what
Tau PET?
The tau PET infancy. So we are now starting to learn how to quantify that, but there's not even yet a quantified cutoff of what a positive tau scan is. I mean, it's so new. There have only been about 6 to 9 papers, a lot of it coming from here, but they've not yet said what is the exact cutoff. So it's still very important to know what the CSF, because there are a lot of work and a lot of work that Dr. Fagan and Dr. Holtzman have done here of characterizing this, and we know this pretty well. We're still in the early infancy of this tau PET. It is coming, but it's not there yet.
Someone else had a question. If somebody has Alzheimer's dementia, how severe can it be, and they still are eligible to participate in our research studies? We only accept people who are mild, not in moderate or severe stage, only mild stage Alzheimer's. Partly, it's because as people advance, they no longer have the capacity to agree or cooperate with some of our studies. For example, they can't still in that MRI scanner. How many of you have had a hard time lying still in that MRI scanner? If people have more advanced dementia, they can't do that. Okay, someone else wanted to know, why the heck spinal fluid? Why not test the amyloid, tau, other proteins in the blood? We're trying. We're trying very hard. There's so much going on in the blood that it dilutes the concentrations of the amyloid and tau protein, makes them almost non-detectable. We're still working on it. Many other people are too. So hopefully it will happen. Then the big question, no, I shouldn't say the big question. A very common question is, what is the difference between Alzheimer's and dementia, and are there other dementing illnesses besides Alzheimer's? All right, dementia. Two things. A person has declining in his or her memory and thinking abilities. A loss of memory and thinking abilities. That's number one. Number two, it has to be substantial loss such that the person no longer can carry out their usual activities. They may still be doing them, but they're not doing them as well as they once did. There's a change. So somebody who drives a car, maybe there's had a change, a loss of memory and thinking. They're still driving, but they might back into somebody at the grocery store parking lot, get a fender bender, or they might have difficulty making a left-hand turn into traffic. They're not doing it as well. That's dementia, okay? Loss of memory and thinking that interferes with a person's ability. Dr. Snyder mentioned this. At the beginning, it's hard to tell whether there's been an interference with everyday activity, but dementia always gets worse, so eventually you can tell they're not doing as well, and eventually they give up those activities as the disease progresses. Lots of illnesses can cause dementia. Lots of illnesses. Matter of fact, once I counted 112. By far the most common after age 65 is Alzheimer's, but not the only one. If you have strokes that affect your memory and thinking areas of the brain, you can have dementia due to strokes. We called it vascular dementia. Okay? Parkinson's disease can cause dementia. There's a frontal temporal dementia. There are lots of dementias. But in late life, the number one cause is Alzheimer's. Now, there's nothing about having Alzheimer's that protects you against those other illnesses. You can have Alzheimer's plus stroke. You may have heard the term mixed dementia. Or you can have Alzheimer's and Parkinson's together. Dementia is the loss of memory and thinking that interferes with daily living. The cause of that dementia most often is Alzheimer's, but not the only cause. Does that make sense? All right, we're past time. I have a very important reminder. Don't forget your coats and umbrellas. Remember, we are a memory clinic here, so we'll be watching you. Thank you very much. Let's thank our panelists. Weren't they terrific?
Get a little bit of recognition.